Hello everyone, I'm going to explain about chapter 6, expression of biological expression in the part of translation. So what's the meaning of translation? Translation means to synthesize protein, where the sequence of nucleotides in mRNA act as a template and is translated into a sequence of amino acids. And we have many types of RNA, which is mRNA, tRNA, and also rRNA. We will go through one by one. So mRNA, which is known as messenger RNA, is a single-stranded DNA which will carry genetic information transcribed from DNA in nucleus to ribosomes for translation in cytoplasm, and the triplet bases on mRNA are called codon. Okay, next is the rRNA, ribosomal RNA, which is the major component of ribosome. I think we all know that the function of ribosome is to synthesize protein, and it will combine with the protein to form ribosomes. So ribosome side of protein synthesis with two subunits, which is small subunits and large subunits. Small subunits contain the mRNA binding site, and large subunits has three tRNA binding site, which is exit site, P site the peptidyl tRNA binding site, and A site, which is translated to amino acid tRNA binding site. Next is the tRNA, which is known as transfer RNA, which is a single-stranded RNA, which is also 70 to 80 nucleotides long. It will fold back to itself to form a compact 3D shape, which is known as a clover leaf shape, as the picture up there, the clover leaf shape, which will held by the hydrogen bonds in between their complementary bases. In the middle loop contains three bases called anticodon, and the protruding 3 and 5 CCA3 ending act as an attachment or template for the site of a specific amino acid. And activation of amino acids is one of the main process too, where the mRNA codon carries the code for amino acids. Anticodon of tRNA has a specific complementary binding sequence for the codon of mRNA, and then we have the tRNA, which will bind to the codon in mRNA, and it will carry the same amino acid encoded by the codon through ribosome to ensure accurate translation happens. And tRNA will bind with the amino acid, which is translated, that matches the code for the correct mRNA codon. Next, the definition of activation is a process in which a specific amino acid is attached to a tRNA molecule to form amino acid tRNA and catalyzed by amino acid tRNA synthase. ATP are adenosine triphosphate, which is an energy source, and there are 20 different types of amino acid tRNA synthase. tRNA is reactivated many times, and amino acids are linked to respective tRNA molecules by the formation of ester bond at the 3 end of tRNA. That's all. Thank you.
Okay, untuk initiation, semua ribosomal subunit ni dia akan bind dengan mRNA. Dia bind dengan specific nucleotide sequence ni. Specific nucleotide sequence ni dia bind start dengan start codon lah, AUG. Ni AUG. Lepas tu initiator tRNA ni dia ada bawa methionine tau, ni methionine. So dia akan bind dengan start codon AUG. So jadi sequence ni jadilah macam yang ni. Jadi AUG ni dia akan bind dengan UAC daripada initiator tRNA. Lepas terdapat macam ni kan, dia jadilah macam ni pula, jadi yang belah sini pula. So large ribosomal subunit ni dia akan attach dengan initiation complex. So dia adalah translation initiation complex. Daripada ni dia jadi sini, dia akan gunakan energy yang dia pakai tu GTP, bukannya ATP. Lepas tu initiator tRNA ni dia duduk dekat P site tau. Dekat large ribosomal subunit ni ada E site, ada P site, ada S site. Tapi initiator ni dia duduk dekat tengah-tengah dekat P site. Dekat S site ni kosong tau, dia empty sebab dia available untuk next amino acid tRNA untuk masuk sini. So bila dia dah masuk sini, dia pindah ke sini sebab ribosom ni dia akan bergerak. So site ni akan berubah lah. Lepas tu dah masuk elongation.
After the initiation state, there will be elongation state. In the elongation state, amino acids are added one by one to the previous amino acid at the C-terminus of the growing chain. In this stage, there are three-step cycle, which is codon recognition and peptide bond formation and also translocation. Let's look at the codon recognition. During codon recognition, amino acid, which is anticodon, which complementary to the RNA codon, enters to the empty site A. What is meant by complementary? Let's look at here. This is mRNA, AUG, and it's complementary to the UAC, anticodon. And the second cycle is peptide bond. Peptide bond is formed between the two carboxyl group of amino acid in the P site and the amino acid of the new amino acid in the A site. It catalyzed by peptidyl transferase. Large subunit rRNA act as an enzyme that's called ribozyme. These steps remove the growing polypeptide from the tRNA in the P site and bind it to the new amino acid on the tRNA in the A site by peptide bond. And lastly, translocation. Ribosomes move a codon ahead from 5' to 3' downstream direction to tRNA in the A site is now translocated to the P site. A site now is empty. Initial tRNA exits from ribosome and the third amino acid tRNA enters A site. Peptide bond is formed between the second and the third amino acid. Amino acids are added one by one to the previous amino acid. The whole process is repeated until all the codons of mRNA is translated.
Termination of translation process occurs when stop codon UAG, UAA, and UGA in mRNA reach at the A site. Stop codon act as a signal, so a release factor, which is a protein, will recognize the stop codon and bind to the stop codon in A site. Release factor add water molecule to the polypeptide chain and hydrolyze bond between last amino acid and tRNA in the P site. This reaction separates the chain from tRNA and newly synthesized protein is released. Next, ribosomal unit dissociate from mRNA and take part in another round of translation. Polyribosome structure. Polyribosome structure are made of ribosome connected by a strand of mRNA that run between the large and small ribosomal unit. Polyribosome appear as a bead on the string, and the bead represent as ribosome and the string is represented as mRNA strand.